Morning children and welcome back to biology class. Today we'll be doing DNA replication and the experimental proof for DNA replication. Okay. Now while Watson and Crick proposed the DNA double helix model itself, they had proposed that if you know one of the strands, we can easily predict the other strand. Or in other words, both the strands of the DNA will act as template. What is that? Act as template to make a copy of its own. That is what is DNA replication, right? Making a copy of its own, right? Both the strands of DNA acts as template for the synthesis of new complementary strand. Okay, which is that complementary strand. Okay, and that too. So in a newly synthesized DNA molecule, one strand is from the parent strand. Okay, and one is a newly synthesized strand. Okay, so in a newly synthesized double DNA strand, one strand belongs to the parent strand and the other one is a newly synthesized strand. Okay, this kind of replication is considered or called as semi-conservative. What is that? Semi-conservative replication. Okay, now the proof for semi-conservative replication was given by Michelson and Stahl. Okay, now what they did was they grew E. coli bacteria in a medium containing ammonium chloride, and that nitrogen in the ammonium chloride is of heavy type. What is a normal atomic number of nitrogen? 14. Atomic mass is 14, right? So which is the heavy isotope of nitrogen? It is the nitrogen 15. Clear? Then so the E. coli was grown in a medium containing ammonium chloride, of which the nitrogen was the heavy type, right? For many generations, these bacteria, E. coli bacteria are grown in that means what? The source of nitrogen for this bacteria is only the ammonium chloride. Why they need nitrogen, children? Why do they need nitrogen? To make the nitrogenous bases, adenine, thymine, guanine, all are nitrogenous bases containing nitrogen. So to make these bases, the nitrogen is needed, and which is the source of nitrogen? Only ammonium chloride, or the nitrogen is heavy isotope of nitrogen. Clear? And for many generations it is grown in this ammonium chloride. Now, this E. coli bacteria replicates or divides or reproduce every 20 minutes. Okay, so as it is, when it replicates, it's using this nitrogen 15, and it is making double DNA. For this E. coli has a double DNA strand. DNA is a double DNA strand. Okay, and it will use this nitrogen to synthesize a new strand. Clear? So after many generations, if you analyze the DNA for its heaviness, for its nitrogen content or whatever it is, you can find that. How do you find this? Analyze this DNA by centrifugation in cesium chloride density medium. Clear? Now what is centrifugation, children? Yes? Okay. So when it is done, both the strands of the DNA of this bacteria will be containing this N15, isn't it? For many generations it is grown in that same medium. So both the strands will be containing N15. Clear, children? Now when it is analyzed, it can be extracted from the E. coli. It can be taken out or extracted and done centrifuge. in cesium chloride density gradient. Clear? And when it is done, because of its heaviness, it may occupy in that medium, on that gradient, it may occupy a certain position. You can look at the textbook. When I am explaining this, you can look at the textbook and follow it, right? It occupies a certain position. Okay, now this group of bacteria or these bacteria are transferred to another medium containing ammonium chloride, of which the nitrogen is nitrogen is the normal nitrogen, which means N14. Okay, right? Now, for this bacteria, this N14 is the only source of nitrogen. Okay, now again, as the bacteria replicates or reproduces every 20 minutes, I told. Now, when it reproduces, it is using this, which one? This ammonium chloride or this nitrogen as the source, nitrogen 14, N14 as the source of nitrogen for making a new strand. Okay, children. Now, after 20 minutes, when this DNA of these bacteria is extracted and again done centrifuge, you can see that it occupies a little higher position than the previous one, because now it contains, now in the double DNA strand, one of the strand is containing the parent strand, which is N15, and a new strand is being synthesized which contain N14, which is now growing in this particular medium. Okay, so N14. So it has an hybrid. All the bacteria will have DNA having the hybrid. One strand having N15, another strand having N14. A hybrid density they have. Okay, so they occupy a little higher position than the previous thing, right? Again, if you allow this bacteria to grow in the same medium, same N14 medium, and again after 20 minutes you analyze the DNA of this E. coli, you can find both types, a hybrid type as well as a lighter one, because some of them will be having both the strand as N14. Clear? And 50% will contain one strand N15 and the other strand N14. Clear with me? Okay, right. You can also still, for another 20 minutes, you can see what will be the density of the DNA of these E. coli bacteria. Another one-time reproduction you can do and see, and you find out. Clear, children? And same type of experiment was done by Taylor and their colleagues. In the plant called as Vicia faba, which is faba beans, a type of beans. Okay, Vicia faba. Okay, in which they used radioactive thymidine. Thymidine is one of the nitrogenous bases, right? Radioactive thymine or thymidine. Clear? And they found similar results. What was the result? That the replication is semi-conservative. What do you mean by semi-conservative now? It means that a newly synthesized DNA will contain one strand from its parental side, from the parent, and the other one is a newly synthesized strand. Clear? One of the strand is conserved. That's why it is called as semi-conservative. Clear, children? Next we'll see how does the DNA replicate in human beings and also in other organisms, right? You know, this DNA is a polynucleotide, a polymer of polynucleotide, such a big large molecule, right? And it is wound against each other in a right-handed fashion. There is hydrogen bonds between the bases, 2 bonds between A and G and 3 bonds between G and C. You know that, isn't it? So it is very and that's why it is more stable. This double-stranded DNA is highly stable molecule, right? And a lot of energy is needed to break the bonds between these molecules. Clear? And that too, the whole length of the DNA doesn't get synthesized at a stretch. A small bit by bit of the DNA gets synthesized. From one side it starts and it just continues and it just goes at the other end. And you should know from which strand it starts, which direction it starts. Okay, it starts from 3 dash 5 dash. You understand what is 3 dash 5 dash and all? We call it as the polarity of the strand. Okay, so the strand with 3 dash 5 dash, in that, on that side only the synthesis begins first. Clear? So there are many enzymes which is needed for this purpose. Okay, from okay, I told you there's a lot of energy involved. Okay, and there are many kinds of enzymes, and if the bond is broken and it is uncoiled from one direction, there is a tendency to again coil around each other. Okay, right? So there is a need of topoisomerase to stabilize that bonds, okay, and to break the bonds between these hydrogen bonds and other bonds, there is a need of another enzyme called as helicase. Okay, these two enzymes what I mentioned now is not there in your textbook, but you should be actually knowing it, right? So the main enzyme needed for DNA replication is DNA polymerase. DNA polymerase, right? Okay, DNA, never forget this enzyme, DNA polymerase. Okay, now this DNA polymerase helps to synthesize the polynucleotide in the direction 3 dash 5 dash. Okay, now first let me tell you. So I told you the double DNA strand starts unwinding from one direction, from starting from 3. Okay, one direction it starts, from one end it starts. Okay, a small piece gets uncoiled and it forms a structure called as replication fork. What is that? Replication fork. Okay, and I told you that the replication begins in the 3 dash 5 dash direction. Okay, so this is the replication fork, children. This is the replication fork. Okay, right, it's this small portion gets unwound and it forms a fork like structure, right? And you see there is a continuously the DNA is synthesized in this strand, the 3 dash 5 dash end. Okay, so we call it as the continuous synthesis, or we can call it as a continuous strand also you can call, or continuous synthesis, because in this the DNA is synthesized continuously starting. Okay, as it is continuing, the next region opens, the next region uncoils. Clear? And meanwhile what happened, the already formed, no, now this is a double DNA double stranded, right? They will start coiling around each other. From one side the synthesis is going on, slowly these regions uncoil themselves. Okay, and meanwhile from another direction it gets already synthesized DNA, right? And it gets coiled. Clear, children? Right. And in the other strand, what is the polarity? It is 5 dash 3 dash. For in this direction the synthesis happen. In this direction. And that too, it is not continuously synthesized. You can see that it is synthesized as small fragments, which is called as Okazaki fragments. What is that? Okazaki fragments. And later, when the replication is going to finish, that those small pieces are joined together by DNA ligase. What is the name of the enzyme which joins the DNA parts, DNA pieces, Okazaki fragments, is called as, yes, ligase. DNA ligase. So in this strand, see, I told you children, both the strand act as template. All these parts should be mentioned in this replication fork. Okay, the polarity should be mentioned, right? Which is continuous strand, which is the discontinuous strand. Both the parent strand act as template. All that things you should mention, right? So in this strand, in this DNA strand, which direction is that? Which polarity is that? 5 dash 3 dash. In this strand, the synthesis is not continuous, okay, and is called as discontinuous synthesis. Clear, children? Right. Okay. So what is the name of the enzyme? DNA polymerase. And deoxyribonucleotide triphosphate. Deoxyribonucleotide triphosphate has a two function to perform, as a dual function. The triphosphate in this molecule contains a lot of energy. Diphosphate plus 1 phosphate, between these bonds there is a lot of energy stored in this molecule. Okay, so it provides the energy for this unwinding and all those things, or replication itself. Okay, and also it helps to add nucleoside plus phosphate gives nucleotide, right? You know that, you remember all those. So to add the substrates to make a polynucleotide. Clear, children? So for you there is need to know only two enzymes names, which is that the DNA polymerase, which helps to synthesize the polynucleotide chain. Okay, and which is the other one? The ligase, which helps to join the small pieces of fragments of DNA in the discontinuous synthesis or discontinuous strand. Clear, children? Ligase. These two you should know it. Clear? See, in this DNA, here it is shown clearly. Okay, so it is getting uncoiled here, slowly getting uncoiled, and meanwhile the newly synthesized DNA is getting coiled around each other, right? And you can see that this strand is from the parent strand, the blue colored, right, is parent strand. The other red one is the newly synthesized strand. So what is this meaning? It is called as semi-conservative replication. Okay, right. So children, the continuous synthesis strand is also called as leading strand. What is that? It is also called as leading strand, and the other strand is called as the lagging strand. Okay, leading strand, the other one is the lagging strand. See, in our case, I told you, from where does it start? Replication. It starts in the 3 dash 5 dash direction. But in E. coli, they have a rounded, they have round shape, not two strands parallel to each other, not like that. They have a ring shaped DNA, which is also called as plasmid, that we'll see later. Okay, it's called as plasmid. Okay, and besides this plasmid, there is also another DNA. This plasmid is the another set of ring-like DNA in the bacteria, some bacteria. Clear? Okay. So in bacteria, the replication starts from where does it start? There is no 3 dash 5 dash end. Okay, but there is a region called as origin of replication. What is that? Origin of replication. So it is in this area, it is from this area the replication starts. Clear, children? But in our case, it is starts from 3 dash 5 dash. Clear? And also you should know that, okay, after each cell division, that is the chromosomes divide and the daughter cells into two daughter cells and all, the cell itself should divide, correct? After the nuclear division, the cell itself should divide into two. Otherwise, what will happen, children? Otherwise, what will happen? The one cell will contain double the set of DNA. And what is this condition called as? Yeah, it is called as polyploidy. It is called as polyploidy. Okay. Right. And also you should know at which stage the replication of DNA happens in human beings. It happens in the synthetic phase, S phase. Children, when after cell division is being taken for you, you will understand what is that. I'm saying what is S phase and all those phases, right? So it happens in the S phase, which is the synthetic phase, right? The previous to synthetic phase, it is the interphase or the resting phase. Clear? And you should also know that the diploid content of human DNA is, what's that? 6.6 into 10 to the power 9 base pairs. 6.6 into 10 to the power 9 base pair. Okay. So it takes nearly 38 minutes. 38 minutes to complete one replication. That is the whole of the DNA is replicated once. How much minutes it takes in human beings? It takes around 38 minutes. And in E. coli, it takes around 18 minutes to complete. That is 2000 base pair per second. Okay, right.